What's up, everybody? Welcome to a impromptu late night live stream here on Logical, Plausible, Probable. I am joined by the one and only Salvador Cordova, and we're going to be having a quick conversation. A few of the people invited, they might also be joining us, but he and I for sure are going to be diving in just pretty quickly into a couple of papers about things that are being done in both information technology as well as biochemistry, genetics, and bioengineering and bioinformatics to leverage the amazing capacities that are at our fingertips when it comes to utilizing DNA for both information storage as well as for computation capacities. And one of the things that kind of inspired this was, a lot of you probably saw the T-Jump versus John Perry debate the other day on Modern Day Debate on is there code in the DNA? And John Perry, who's an atheist, I felt like I was listening to myself in his presentation, his arguments. It's like he's outlining everything, like here's why it's code, here's what computer code does, here's why it's exact corollary, all these different things. And of course, T-Jump and a lot of the atheists continue to, scream that it's not really code. And then John Perry, we actually great after show. John Perry came on, we had a great conversation, and we agreed on all these kind of things. Of course, in his context, he thinks that signal theory somehow explains the formation of code and its expansion, which I think is just ridiculous because that requires completed systems in order for any kind of modification or addition to the system. But that's a whole other conversation. That aside, I wanted to, I was kind of like perusing through a few papers, came across these ones we're actually going to go through tonight. And I was looking for some graphics to explain like how, some diagrams of how data is being stored in DNA. And then I'm like clicking on some stuff and I'm like, oh, here's a new paper I haven't seen before. Oh, here's a new paper I haven't seen before. And I quickly, I'm kind of perusing through these and I'm like, wow, this is, I knew a lot of the stuff that was already being accomplished, but I'm sitting there reading through these different publications and like, wow, that's pretty awesome. So I said, hey, why don't we do a quick live stream? Hit up Sal. He said, yep, I'll join you. So here we are, ladies and gentlemen. So Sal, thank you so much for doing this on the fly. Oh, and by the way, Sal also has, he released a short video, a 3-minute video on his channel a few minutes ago, right before actually I asked him to do this one, on the racemization dating algorithm our formulas he came up with, that he was kind of like, hey, speed of sound, here's the math, go create your program, go create your script in Python to double check my math here, which we all know that's what he's trying to do. We all know that Mike just wants to try and find a way to, debunk the whole point that Sal and Dr. Carter were making the other night about racemization dating. And he's going to have, I think, correct me if I'm wrong on this, Sal, but I think you have that was a 3-minute video. I think you've got an hour-long video where I'm guessing he's going to like break it down for everybody. But
yes,
what they can expect and should be looking out for.
Well, I'd like to thank Mike Riesenack in advance for doing free of charge peer review and independent verification and validation. And we're all kind of joking. He said, what if he finds out that I'm right? Is he going to keep it to himself? So
it's a very good question.
What I try to emphasize to Mike, you're trying to make this about me. That's not the issue. Why don't you go to the peer-reviewed literature, find out a formula for K. I gave him my version of it. He ought to be able to reconstruct it if he really wants to. And just set some parameters and make K a function of T. It's really easy. It's not that hard. Anyone who claims that degree of knowledge that he does ought to be able to do this. And, trying to make it about me that's just not cool.
Well, and I think one of the things that the atheists need to start recognizing is I personally am done with you present evidence, and if it has been touched by somebody who believes in intelligent design or creation, or both, therefore, from their perspective, it's tainted, and they're just going to dismiss it by default. And it's kind of tell you what, I think this is kind of the point you're making. Go look at the secular data. Go apply the logic yourself. and then you do it. I don't not know. You're going to sit here and try and make there's something wrong with your formula and your spreadsheet, right? I mean, I can already envision that being the excuse. And it's like, no. Go write your own script. You input the math. You go and put the formulas, and then see what happens. Don't just don't make up claims that my data is incorrect or my interpretation is wrong. So that's
Yep.
Oh, he's already talking trash in chat. So.
Oh, he is.
Yep.
Okay, Mike. I made a 3-minute video for you. You ought to be able to do this. You don't have to make it about me or anything I said. You can go to the literature yourself. Make K a function of T. Keep DL constant. Tell me if you get that result. It's that easy.
And Danny, we are not abusing speed of sound. But
it's not that hard. I gave you a formula. I presented it in 3 minutes. I presented the formula in 30 seconds. You ought to be able to do this. You can build the spreadsheet yourself. I don't know why you're just trying to trash my work. Don't make it about me. Make it about the truth. And what are you going to do if you find out I'm right? Are you going to keep it to yourself?
Well, I guess we'll only we'll have to wait and find out and see what.
We'll see. We'll see what this graph does. I've lost my patience because speed of sound. You're supposed to be a friend. You've been trash talking me privately. You've been talking about kicking my ass in emails. I blocked you briefly because of that. You said I need a brain or something. That's just not cool, man.
Ooh, okay.
That's just not cool.
I would agree, man.
Recently, and then you trash talk me after I take time to re-deride the formula and spoon feed it to you. That just isn't cool, man. That just isn't cool. So
okay. Man, he's now getting he's taking it to a new level. Actually, I publicly trashed you, is what he just said in comments. Well, speed. We're not going to make this one all about you.
Yeah,
it'll be interesting to see if you can actually create the formula. You talk a big game. You talk about how people like Sal can't do the math, can't do the analysis, aren't being able to do full scale, deep dives into the data, and it's all biased, and the conclusions are biased, and all this kind of thing. So, the point that I'm trying to make here is, a lot of people have given me grief about how I approach a lot of these things with the aggression that I do. And it's, what? I'm tired of you guys talking trash and not backing up your BS. And we're backing up ours with data. So just because atheists want to live in the atheist fantasyland projection complex, fairy tale land, and which, by the way, folks haven't seen it, there's a video on the atheist fantasyland projection complex, along with the droid C, which is the desperate rejection of intelligent design condition, which usually goes in conjunction with the AFLPC and creates an exacerbated problem. But let's get back to this whole conversation we're doing tonight, which is there code in DNA? And in this can binary code be translated into quaternary code, which is into quaternary, which is what the genetic code is in. But there's so many different, what I find very interesting, and I'm sure, Sal, I would wager you do as well, is, a lot of these things that the research is being done now that we're going to kind of showcase in some of these papers is it's built on the acceptance that information and communication theory and those principles that we apply to information technology and telecommunications and everything else in that arena, they're just accepted by all these people doing the research, right? Like, yes, these all apply. These are 100% correct. These are directly applicable to genetic information. Like, they're not arguing about that at all. They're looking at real world practical application of it and having their mind blown at the possibilities. Would you say it's a fair statement?
I think it's pretty close. It's pretty close. Now, I'm always reserved about this because the other sauce to make it, and it's like, okay, we don't make it an issue. Just tell us the probabilities of these systems arising that are able to do at least something that looks like this. I mean, you don't even have to prove that it's a code. Just say, gee, it really looks like it looks like a code. It behaves like a code. What's the probability of making something that close to it? And that's usually sufficient. But this is the sort of argumentation that one goes to when one is running out of arguments. So I kind of like stopped watching the debate when T-Jump said, yeah, ideas aren't real things. I'm like, okay. And then John Perry said, okay, so what's in your head's not real, or something like that. I was like, I said, that's all I had to hear. So, I mean, what do you do with that at that point, you know?
It's reaching levels of just full scale refusal to accept the obvious and just concede. Like, hey, come up with a good explanation for it. You know, go run down your signal theory rabbit hole, you know, and your frozen accidents, because that's where you're at now. I mean, and I tell you, sidebar, something I'm actually going to ask, you may be able to expound this. I may see if Dr. Carter wants to have like a more in-depth conversation about this. But that whole point he was making about the hook on the tRNA to only expose the required 3 nucleotide bases for the codon-anti-codon pairing. I found that extraordinarily interesting. And actually was looking kind of at the 3D structure of the tRNA earlier, and sure enough, there it was, this little weird, at the very bottom of the spiral, like this little hook. And there it is, 3 base pairs hanging out right there, the 3 nucleotides hanging out right there for the match. And I'm like, that's just amazing. You know, oh, and we have all these ones that just happen to have the appropriate codon-anti-codon match. And then at the other end of the tRNA, you got a whole nother set of customizations that are required in order for it to be charging with the appropriate amino acid. I mean, that whole premise to me is just it's amazing on so many levels, but just the premise of that from a translation of information perspective is pretty cool, in my opinion.
Now, I saw some people tagging me out of respect for LPP. While I'm on the show, I do try to focus on my co-hosts and other people, and I'll focus on you and your questions. It's not to.
Oh man, we let's all like. I've got no problem with you responding to people on chat. This is a late night impromptu. We can have some fun on this one. Okay, so without further ado, folks, let's get into some of the stuff. So one of the first ones I'm going to show is I'll pull it up on my screen here in a second, but it's titled "Robust Chemical Preservation of Digital Information on DNA in Silica with Error Correcting Codes." Okay, so for those of you who are going to be ignoring the obvious, the title, "Preservation of Digital Information on DNA." Digital information on DNA. Okay, it's really obnoxious listening to YouTube atheists try and act like there is no digital information that is being stored in DNA. The bases are the digits. Like, this is what it is. And if you refuse to, it's like they are the 0 and 1s, folks. This is what's happening. It's quaternary versus binary. Like, this is a well-established premise. And all right, so I'm going to share my screen, and I actually might post a link to this on YouTube. Okay, so zoom in on this a little bit. You'll be able to see that in okay. All right, so what these guys did is, I'll just read some real quick here. So Here we show that digital information can be stored on DNA and recovered without errors for considerably longer time frames. They're looking at this in terms of like how long can you store digital information, and right now they're having like anything like the sense for. Okay. However, the storage of digital information for time frames exceeding 50 years is challenging. Here we show that digital information can be stored on DNA and recovered without errors for considerably longer time frames. To allow for the perfect recovery of information, we encapsulate the DNA into an inorganic matrix and employ error-correcting codes to correct storage-related errors. We specifically we translated 83 kilobytes of information to 4,991 DNA segments, each 158 nucleotides long, which were encapsulated in silica. Then they did different aging experiments to kind of figure out the decay rates. Okay, so let's. I'm looking at the wrong thing here. Where did I store that? I'm on the web page, not the. Oh, there we go. I shared the wrong tab. Hang on. Okay. All right, so as y'all can see right here, they've got a diagram. It's going to be an illustration of what is going to be happening. So you've got binary code right here. The encoding process is happening where it's being turned into DNA code. Then you have the DNA synthesis, creation of the actual strand, encapsulation, and now we're going back. So we got the encode, storage, translation, or the re-encode, and we're sequencing it back all the way back into binary. This is a very short, simple explanation of what is going on. So just case in point, I mean, to be honest, Sal, I think we could probably just end right here, right? Like they're literally showing the process by which they turn binary and the data and the information that's stored in the binary code into genetic code or DNA code, and then retranslated back into binary. I mean, I don't know. Like I said, correct me if I'm wrong, but I think we could do a mic drop right now and just be like case closed.
What I'd like to say is we sometimes use shorthand words to describe things, and a little bit of modification of those words will kind of insulate us from, cheap shots. So I've often said DNA is a substrate that's used to store information. That's the more correct thing. If we say DNA is information, that's accurate enough, but it's not. It kind of makes it vulnerable to cheap shots. The reason I say that is DNA is a lot of the non-coding DNA. It may or may not have a code in it, but it has very important structural properties that make what they call the 4D nucleome. So DNA is actually polyfunctional. It's not just used for information storage. It is used as a structural scaffold. It's like a ladder or a parking lot that can also store information. It's multi-purpose. It's a multi-purpose chemical. So,
well, and I think I agree with everything you're saying there. I think one of the components that needs to be addressed in line with what you're saying there is, I don't know how many times people have to, like myself, like you, Otangelo, say we are talking about the genetic code, which is stored in DNA. Physical substance stores it, in line with what you're saying. Now, the point you're making is that, yes, while it is the physical substrate, it also has all this other functionality that's required for the expression and the access to the data.
Yeah, especially in humans. What they had presumed that for DNA to be useful, it has to be only used as an information storage system. They didn't take into account that it can actually act as parking lots. And in the chromatin formation, it's part of the physical hardware that enables the chromatin computer to work, where the DNA is actually host to a random access memory. And that's a whole nother dimension. And that's what they've been missing.
Well, and
It's
it's actually
actually because they were saying DNA is code only. It's like, no, it's not. It's hardware for a random access memory. It's not just a read-only memory.
well, and it's interesting. Actually, hopefully I can find it again. I forget which paper it was in. But one of the papers that we're going to look at tonight actually talks about the random access memory portion of it. They had a little subsection on that. Being able to utilize it for RAM as random, well as ROM.
Exactly.
And that to me, when you really start to think about that, it puts so many things into context, number one. But number two, one of the other things, we may not get into this, one of the other things that's mentioned in these, actually, I think in all of these papers, it's mentioned that the rewrite function right now, human ability to rewrite DNA is extraordinarily slow, right? And to do modifications, like it's extraordinarily difficult and slow, right? To me, when you think about that, it's like, okay, people, let's use our brains for one moment. It can be done, but it's really hard, right? We have a ridiculous amount of data that's stored in our DNA right now, which enables life to exist. So you're saying that data that is stupidly hard and way more advanced than what we're currently attempting to store in genetic code right now. You're arguing that happened through undirected process, like environmental pressures and natural selection somehow created all this high-level data and functional information when we're having an extraordinarily hard time. Like, it takes us a day to store a megabyte. Like, come on, folks. Let's think about this for a second.
Well, if I could relate that to the work of Dr. Tan, she said when Craig Ventnor was trying to make this artificial life form, where he said the parent is a computer, which isn't true. That's just a tremendous exaggeration. They were taking the computer bits and then trying to synthesize DNA. In your first article, they said they had the 150 base segments. The reason for that is we have a very hard time making very long DNA segments. Mm-hmm. Cells can do it easily. And in Change Tan's book, she said, this is so hard to do even in the lab if you're trying to build it from scratch, where you don't have a pre-existing DNA template. In living things, you have a pre-existing DNA template that makes it really easy. To be able to take it and build it from scratch, like say you had the bits in mind or the bases in mind in a computer, and now you're going to try to translate it into chemistry. It is so difficult. She said that's evidence against the naturalistic origin of life, because you need so much machinery just to be able to get one base to connect to another in the right orientation, because there's so many orientations where it can go wrong. That's why it's so difficult. And so what ends up happening is you put one base, you have to have all these enzymes and chemicals to put it in the right orientation. So you want to put in an adenine. Then, the next one is going to be a cytosine. Oh my goodness, this is really hard. We have to pull out all the chemicals in the assay, pull it all out, put in all the new chemicals. Now we can put a C. And now we want to put another letter. Okay, you pull out all the chemicals. You have to stop the reaction, pull out all the chemicals, put in the new chemicals, restart the reaction, and then stop it, because you don't want it to keep growing. And This is brutally painful, and it's error prone. You have to go back and check that you got the sequence you wanted. And I'm glad they're up to about 150 bases, but what Ventnor had to do is he had to take those 150 bases and then stitch together using a biological organism. This is brutal.
Well, and that's the thing. They're also, these guys that we're reading these papers on, they're having to do the same thing in theirs, because they're utilizing like their read process is read and write is utilizing natural enzymes that they just learned how to control, which is the operative piece
So I mentioned the read-only memory. So we normally think, we actually said DNA is a code. That is kind of in the context of read-only memory. What we found out is it's also a substrate to help implement epigenetic random access memory that is tied to the histones and then methylation states of the DNA. So that totally changed the picture. And that's what kind of changed the view of quote unquote junk DNA. It's like, oh my goodness, this is a massive random access array. It's also parking lots for molecular machines at the same time. And we began to look at DNA as part of a three-dimensional storage system, which we don't know how to do.
Well, and
We
here's
tend to think of storage in only kind of one dimension along the sequence. The human DNA is so sophisticated, they're not saying it is actually a three-dimensional storage system. We don't really understand how it works. I have papers on that. So all this to point out, it's a nice shorthand to say DNA is information, but I prefer to say DNA is a substrate that can be used to store information, because it can be used for all sorts of things beyond that.
Well, and that's one of the things that we're going to touch on later on, is the whole aspect of it looking at it as a computational substrate. And and for the simultaneous computations that could be being executed, and the ability to replicate on the fly, to go down two different computational pathways. That kind of stuff to me is one, pretty nuts. I'm talking from a very cool perspective, but two, like, okay, folks, do you guys not think that there's computations like that happening right now? Like, how do you think we exist? Like, how do you honestly think that the most advanced nanotech robot? Sidebar, folks. Sal did a short, I think it's about 12-minute video earlier today, actually, on, I forget
the cell is a machine.
The cell is a machine.
Response to Speed of Sound saying Sal has some explaining to do because he said the cell's a machine. And I'm like, no, I don't. One of the top 20 medical researchers, he published a book, Cambridge Biochemistry Library. He said the cell as a machine, and he taught a course on it at Columbia University, one of the premier schools. I'm like, no, I don't have to do any explaining.
Well, and that, as a sidebar to the audience and continue on the conversation on this topic, I can't tell you the number of academic conference lectures that I've watched on YouTube where the PhD in X, whatever topic, gets up and does a full-scale presentation about the nanomachine functions of proteins and different systems that are happening inside the body. And they literally, the part that cracks me up more than anything, I remember one of them, I'm having a brain freeze on the guy's name, but he started his presentation with, I'm paraphrasing, but it was, I know people say that it's, analogy, metaphor, whatever, for when I call it a nanomachine. No, it's a machine. that was the part of his intro. And then he goes off on a whole exposition about why it is and showing all the different pieces of it. And I was just cracking up. And every time I listen to the YouTube atheists claim that it's just an analogy, I'm like, well, the guys that are doing the research are saying the opposite. They're being like, no, for all you idiots out there who think it's just, chemical reaction, whatever. No, it's a machine. It's nanotech. We're trying to copy this and make our own nanomachines that can do this sort of function. So if we build it, it's a machine and it's nanotech. If it already exists, it's not. What?
And the thing about Dr. Michael Sheets, he said the way to understand the cell is to reverse engineer it. You have to look at it as a machine, and then you reverse engineer it to understand the relationship. First of all, to understand the parts, and then the relationships of how the parts work together. And very few biochemists that I know of, except people like Larry Moran, will say that the cell is just piles of junk. The things that I have read, especially the later literature, they say this is exquisite, this is sophisticated, it's artful, it is masterful, it is clever, it is crafty. These are from peer-reviewed articles, and I've read it in my journal club. It's like, why are they saying that? Where do you hear that it's junk? Does it come from the people that actually do the research? at the cellular level? No, it comes from evolutionary biologists and philosophers of biology.
Now, ladies and
People who
gentlemen,
don't know, it's not from the people that actually build it, and also the people that do genetic engineering. It's like, wow, the way it does it in the cell is so much better than we could do. You gave an example. The cell can replicate DNA really quickly.
2 meters. I think you mentioned earlier 2 meters in about an hour.
Yeah.
I mean, and you put that in
space, it's like 5 microns.
Well, and that's the thing, Sal. In terms on the information, I can't remember if you were on the stream when I went off on tangent about this the other day. In terms of data compression.
Yeah, I heard it. Now, I didn't quite agree, and that was, it's like I was too tired to talk. It is physical compression, and I'm fine with that. Now, there is a form of data compression in DNA that's very fascinating, which I'll tell you.
Yes.
All your cells are basically lossy decompressions
Yes.
of the original form, because when you see that each cell is slightly different than the other, we also know that each DNA is not exactly the same per cell. We don't quite understand it. There's a few enough changes, particularly in the brain, and I recognize it as a lossy decompression, which means DNA it's compressed in a way different than the way you described it, but it is highly compressed. In fact, we don't know how to make the sort of lossy decompression that the DNA does, because if you think of all the cell types in a human being, and the DNA is slightly different, particularly in the brain, and this is one thing that was shocking. Every brain cell is unique, and its DNA is likely not unique, is unique also. That all these long intergenic non-coding DNA,
the LNC
lines,
the LNC RNAs or whatever.
No, it's called the line.
Yes.
Yep. What they do is they rearrange the DNA slightly among the brain cells. It's not the same. It's not the same sequence. This is very important if you think about it. How does the brain, if you want to store information like in the brain, you have to have unique addresses to store the little bits and bytes. Mm-hmm. And the brain has to create all these, maybe 100 billion addresses just among the neurons, and it has to change the DNA. And so how it is able to do this in an organized fashion so that it's...
Well, and here's the other thing on that point you're making. Not only does there need to be addresses in order to access the information, in my opinion, where all of this research is leading is it's not plausible for us to be being able to access this information as fast as we do and as efficiently as we do purely through chemical reaction. Like I'm not saying there isn't chemical reaction. I'm saying that there's got to be an additional process going on somewhere in order to be able, to be actualizing the data as fast as it's occurring. To me, I'm like, there's got to be there's an additional variable that we aren't even aware of yet that's going on.
Yeah, there are things that happen so naturally to us we don't even give it a second thought. You recognize someone's voice. That's tremendous amount of data retrieval right there. Yes. And for people that worked on voice recognition and speech recognition, it's like, oh my goodness, this is like non-trivial.
Well, in line with that, anybody who has an Android will know this. About a year and a half ago, Google, with Android, they rolled out their new attempt at originally Google's voice to text was based solely on pattern recognition and like translation of it's like I'm literally measuring if this sounds. Obviously, they're doing the math, the amplitude, and all that kind of stuff. But does this sound like X word? If so, then apply the variable, the probability calculations and go, right? Then they tried to roll one out doing AI projection for like grammar rules and things of that nature, and voice to text went absolutely haywire. You get a period, you didn't say period, and it would like give a period in the middle of a sentence. Crazy stuff, right? So they reverted back to their old one. Well, about 3 months ago, the idiots at Google rolled back out their stupid AI autocorrect voice to text function, and it has been driving me up the wall. It's like there will be words that get inserted that are obvious attempts at context or to like they want to play on the grammar rules, but phonetically, the word they're inserting is not even close to what I said, right? So it's like, it's not that they're misinterpreting or I'm slurring the word or whatever. No. It's not even remotely phonetically close. So the original, process I was talking about is obviously trying to be mitigated on their end, and they're trying to apply AI to predict what it's supposed to be and what I would probably have said, right? It's like, well, hang on, that's kind of weird, and you're failing miserably. So the point you're making of, some of the highest caliber tech heads on the planet have been trying to pull something like this off, and they're failing miserably, and we are able to do it at an extraordinarily high level, and we can also carry on multiple conversations simultaneously, right? Like, we could be comprehending something on TV while having a conversation while sending a text message, right? And we even have additional, while listening to music. I mean, there's all sorts we can have multiple layers of this going on, while also feeling physical sensation and that data, right? I mean, and temperature, all those different things that are being calculated and computated at the same time. And we just gloss over the fact that what the heck's having to occur in order for this to take place.
Oh yeah, it just seems so natural. One doesn't realize it till you try to build a system similar. Change stance, understand life, try to build it. Then it's a different story.
Yes.
And that's how she teaches her molecular biology course. We're going to try to understand life by building it. Yes. And by the way, I'm trying to record her molecular biology course. I was distracted today. I wasn't able to finish, but like the first segment. So she will. She tells me that she is planning to sit in and listen to Dr. Carter and I on Sunday night talk about the improbability of eukaryotes. And I'm also going to argue why the nested hierarchy fails as an explanation for eukaryotic evolution. So that's just a little self-promotion for my channel.
Oh, awesome. That's going to be Sunday, right?
Yeah, Sunday night. And hopefully it'll be a 1.5 hour lecture. We're going to talk about intracellular transport. And we're going to show why I'm going to try to argue why it's ridiculous to be arguing the nested hierarchy solves it. It doesn't, unless one invokes non-random mutations and that are rather miraculous. So, yes, your diagram. Go ahead and show it.
No, that's great. I'm actually very much looking forward to that. I think everybody should just put a checkbox in your notifications that you need to go watch that, because I'm sure that's going to be a very high level conversation that is worth your time. Okay, so getting back into...
Thanks to Doki for helping me out.
Yeah, appreciate you, Doki. Awesome on that one. Okay, so this portion of a little expansion, if we're talking about the redundancy, the original information, the packaging, creating the data packet. So you've got adapters, you've got the 107, they're showing their diagram here, 117 nucleotide long encoded information. You got another adapter at the other end. Now you've got the DNA sequence, the decode of the intercode. You got sort and remove indices. Do we ever have that in natural systems? Sort and removal of indices?
Oh, look at that.
Do we?
I just saw something. I mentioned Reed-Solomon encoding.
Yep. There we go.
Here it is, Reed-Solomon.
Yep.
That is so cool. That's part of it.
And here's the other thing, Sal. In one of the other papers, and this is actually discussed in quite a few other papers I've read, they talk about the equivalent of like Hamming codes and Reed-Solomon codes. I mean, there's different stuff. Like we're trying to implement it, but then like basically natural systems have their own version, and it's like we just don't know. We know it's there. They just haven't been able to decipher it. They haven't been able to, as the point you were making earlier, they haven't been able to reverse engineer it. They know it's going on, and kind of have some ideas about what the natural error correction codes are, but just haven't completely determined what they are. It's crazy.
And just briefly, the reason I mentioned Reed-Solomon encoding, it's really amazing. I'm working in biology now because my primary training was in engineering, and Dr. Sanford hired me. He said, "You had the right skill set." I'm like, "What? I don't know anything about biology." He said, "No, that's the right skill set." And I later found out that a lot of his research teams, like say at the NIH or other institutions, they don't hire just pure biologists anymore to solve this. They hire teams of computer scientists, physicists, and engineers and chemists. That's just the norm now, because it takes a team with that diverse skill sets to be able to figure out how things work. And so I was mentioning regarding the Reed-Solomon encoding, some years, 2 decades ago, some biologists were giving a hard time and saying, if God were a competent designer, he'd do read-write operations and it'd be perfect. There'd be no errors. And I saw right through that. I said, you guys are idiots. You just don't know what you're talking about.
And the irony of ironies,
Yeah.
an irony of ironies, to expand on your point, Sal, and I know you know this, but for the audience, there is a plethora of papers that literally are titled things such as read, write, and erase enzymes for epigenetic controls. I mean, different things of that, methylation and things of that nature. Like we have now fast forward to the present, to prove your point. We now have determined or being able to analyze which enzymes are operating as the read, write, and erase functions that you're talking about, and they were claiming would never be there unless it was designed. And it's like, well, here it is. Congratulations. Are you going to concede now that there was a designer? Of course they're going to deny it, right? Oh, it evolved. It was some signal theory.
And so one thing I learned, I said that was Claude Shannon, who invented the word bit and became very famous. By the way, his colleague was the guy who invented how to beat blackjack. He's also a great mathematician. Shannon, a consequence of Shannon's theorem in his famous paper on communication,
1948.
Yes, was that there exists a error correction algorithm. You can always build an error correction algorithm that if you have a certain signal to noise ratio, meaning that there's errors there, you can reconstruct the original signal correctly. And the consequence of that is it's actually better to create a CD that writes with errors and reads it off with errors, and then you correct the errors on the fly, than it is to try to write it error-free. Because what kind of CD would you have? A CD with maybe just 1 or 2 bits? Because then it's going to be error-free, but you don't have any data capacity. If you want to maximize the data capacity of your CD, you read and write with errors and you correct it on the fly. Shannon proves that there should exist a error correction algorithm. He doesn't tell you how to build it, but it should exist somewhere where you'll be able to reverse the errors with high fidelity up to an arbitrary, any desirable level of error correction. And that was the big breakthrough. And so these biologists say, oh, that was such an incompetent designer making error correction. He should write it in the first place. I said, you guys are idiots. You have no clue what you're talking about. That's why you don't make disk drives, because you don't understand the principles of communication theory and information theory. You wouldn't be saying crap like that if you knew what you're talking about.
In irony of ironies, Sal, one of your favorite people of all time, Dr. Danny Boy, on one of my streams, not Dan Grauer, the other Danny Boy, on one of my streams a couple weeks ago, he stated that in evolutionary biology, and for him as a PhD, knowledge of information theory had no bearing. And he freely admitted he didn't know anything about information theory. And I'm just like, then how are you teaching this in the modern era? Like, how do you get a PhD in genetics and virology if you don't know if you, per your own position, you claim it has no bearing on biological systems? Like, that means by default you are an idiot and are not going to be able to comprehend the macro view. You're going to be blind to the fundamental foundation, the recognized fundamental foundation for biological systems, like as we're showing right now, right? This is a foundational premise, and you're claiming that you don't, A, it has no bearing, and B, you don't need to know anything about it, and C, you don't know anything about it. So to me, that is like a physicist saying I don't need to know jack diddly squat about general and special relativity.
Well,
And they're being like, well, I'm an expert in this. Like, whoa, what? You can't hold the phone, bro.
Well, what's happening is that for the sort of work biologists have done in the past, that was adequate. But to really take it to the level where we really understand how organisms work, what we're finding is no one is going to have all the specialties. All of our research teams, there's no one that knows it all. It's unbelievable. When I see papers now, they have like 10, 20 authors.
Mm-hmm.
And there might be one guy on there who understands information theory, because then you need the chemist who doesn't understand information theory. Then you need the geneticists, and then anything else that's there. So, I'm sort of saying, let's go easy on him, because that's just the way biology is so complex. No one's going to know all the fundamentals. We're going to miss it. And
yeah, but in this particular topic, I agree. You don't have to be an expert, and nobody can be an expert in everything, right? I agree that you don't need to go out and be like some crazy, like high level, being able to do the stupid levels of math that are involved in, especially on the Shannon information side of the equation, right? Like that, the math on that is nuts. I'm not suggesting that the biologist needs to be an expert in that, but
okay.
I'm saying that they need to understand the principles.
Yeah,
and just the basic, because I don't claim to be an expert in it, but I know the fundamentals of basic information theory and basic communication theory. I understand the fundamentals, and therefore it enables me. I'm speaking for myself. It enables me to comprehend a lot of this stuff, because I'm like, okay, I don't necessarily know all of the intricate details, and I couldn't necessarily go and recreate the math, but I understand the premise and the implications that are being put forth and why they're relevant.
Well, I'll give a very simple example. The DNA has 3.2 gigabases, which is 6.6 gigabits. If you take 8 bits per byte and you say the genome's 90% junk, and you do some calculation, about 80 megabytes. Do you think that's enough to make something as complex as a human being? Simple math. And I'm just like, it's really presumptuous to say 80 megabytes is going to make all the systems.
I'd be willing to gamble that more than 80 megabytes is required for the functional capacity of a drone, of the most simplistic drone. Would you say that's a fair assumption?
Yeah. And that's why I went into this non-DNA inheritance. I said, there has to be other places the information is stored. You can't make a human as something as complex as a human, even if you assume 100% DNA functionality, and you have, that would be what, 800 megabytes? Do you think 800 megabytes, you ask engineers, do you think that's enough to make a human, something as complex as a human? And so that's why I said I had that show on non-DNA inheritance. And I think I proved by citing all the experiments involved that a lot of information is stored. In fact, I didn't have a chance to go to the papers that talked about the glycome, and the glycome dwarfs DNA. Maybe the most information storage is in the glycome, in the sugar code. And we were only scratching the surface. The only reason we focus on DNA, it's human nature just to focus on things we understand and try to define how everything works in terms of what we understand. And maybe the answer is something that's beyond our reach at the time.
Well, and you've heard me say this many times. I think you agree with me on this. We don't even know what to look for.
Yeah.
And I think that is freely being, that premise that I'm putting forth right there is being proven beyond all doubt. By this manner. With all of the, and I'm having a brain freeze on the term. What's the term where they like look at biological systems and try to copy it and create? It's like
biomimetics.
Biomimetics. The one, we're doing that, but secondarily, I'm speaking this specifically in information technology, right, and robotics, drone technology, things of that nature, right. Up until very, very recently, we didn't even know what to look for in terms of the functionality, right, that's required in both from a physical function, right, the physical widget, as well as the information, because we didn't even know that kind of coding was possible, right?
Yeah.
We didn't know what to look for. Now, as we're trying to write very dumbed down versions of it, people are now recognizing, oh, I wonder if that's what's going on in a biological system. And then they go and look for it. And what do they keep finding? Oh man, turns out that there is XYZ type of computation occurring in a biological system, which enables XYZ function to occur, right? So I think that's just going to continue to scale as our own information technology, robotics, et cetera, continue to expand and improve. Somebody's going to have an idea of like, I wonder if we could do that. And then some other guy's going to hear about it when it comes out and go search for it and find it. And but to me, what does that prove? Either A, random mutation and natural selection is the greatest engineer of all time through undirected process and environmental pressure, or you have to have a super genius. And to me at this point, when you're getting into things we already know about, if you haven't already reached that as the more likely conclusion, I don't know where you're at. But okay, so speaking of that, let's get okay. So this next paper we're gonna pull up is entitled "Nucleic Acid Databases and Molecular Scale Computing," which I think is a very interesting topic. This was actually published last year, I believe. Some guys at Duke University. So not exactly. It's their irony of ironies. You might like this now. The primary department is the Department of Electrical and Computer Engineering. The subset is the Department of Biomedical Engineering. So apparently, the electrical and the electrical engineers and the computer scientists have been recognizing this. So get this, Sal.
Oh yeah.
They've been,
oh yeah.
Look at this: in vitro DNA computing and they actually part of this paper's topic is they've been limited to in vivo. Now they're being able to do in vitro computing, and with DNA, and which I thought was kind of cool. But they're looking at different ways to do both and like find out which things are the most efficient manner. So you can see here like in vitro versus in vivo. I thought that was kind of interesting. But they're really getting into, oh, and by the way, keywords. Check this out, Sal. The keywords: DNA, nanotechnology, synthetic biology, digital data storage, molecular computer. I mean, cellular memory. I mean, folks, at what point are you YouTube atheist, like blind as a bat people, going to recognize that people like myself, Sal, we're not making this stuff up, folks? Like, it's here and it's real, it's reality, and you guys can either accept it or live in a fantasy land where it's not real for you. Okay, so the next topic. So enabling technologies and techniques, long-term preservation of DNA. So I'm going to look at this both from a chemistry perspective as well as a information technology. Look at different ways to get this DNA sequence design. Oh, by the way, sidebar. In that course that you did a little brief overview on earlier, you did the word search for evolution and found zero. In these papers, folks, if you actually go through and read them, you will see the word design everywhere, because turns out they're having to do crazy high levels of design to be able to pull this stuff off. Okay, targeted editing. I mean, what was one of the biggest problems? To do data storage, we have to figure out how to insert the freaking data. So they're coming all sorts of ways to do that. But now here we go, folks. This is the part I really want to cover. So DNA computation and databases. I mean, it's crazy stuff, but okay, just as an example, tools such as Visual DSD can prototype, model, and analyze DNA-based reaction networks capable of solving sophisticated mathematical functions or implementing digital or analog circuits. In particular, the toehold-mediated strand displacement mechanism has led to both diffusion-based and spatially localized DNA computing systems. I mean, I don't know how much more direct you have to be at this point. I mean, we're creating DNA-based computers, and they're realizing that they have the capacity to be more practical and be able to do greater amounts of computations than quantum computers. I mean, that's pretty nuts when you think about that, the potential capacity. And if you read this, but folks, if you read this paper, I'll put a link to the folder for folks want to read about it. It's great. The stuff they get into is absolutely nuts. Now, originally they were talking about computation. Now they're talking about DNA databases and what they did. But here's where it gets kind of fun. So in 2012, Church et al. Used synthetic oligos to encode data containing a book, 11 images, and a computer program. They split data into chunks to avoid long sequences and encode only 1 bit per base to mitigate undesired sequence patterns. An address was incorporated in each oglio to order sequence assembly. However, this approach could not support random access, as the entire pool must be sequenced and decoded to retrieve any file. After amplification and consensus alignment, the authors recovered the original 5.27 million bits with 10-bit errors. Goldman et al. Stored various file types, ASCII text, PDF file, JPEG picture, and MP3 audio in their DNA storage with error correction. Using a customized Huffman code, they translated each byte into a series of 3 base digits and were converted to nucleotides via simple rotating code to avoid homopolymers. I mean, of course, they go on to expand the on what they did, but I mean, at this point, they're storing MP3s, folks. Like, and they're being able to extract it back into binary. And of course, the rest of this goes into different experiments they're talking about they were done and how they've improved it, reduced the error rate, things of that nature. But I think this paper had a very interesting diagram. While I'm looking at this, feel free to inject anything in the conversation, Sal.
I have a couple thoughts. One thing is there are more levels to DNA than just as information storage.
Yeah.
It's what they put in that is compelling. And I have two. Let me see if I could find. I have 2 examples here. First, at the protein level, we were talking about just the levels of computation. You're saying that we don't even know what widgets to look for. That's true. Now let me show, I'm going to share my screen here and show a cryptochrome protein. Now one can look at this and say, hey, how long is this? I should know the size here. Let me check on the size.
At the low end, I'm going to guess that's a couple, no less than 1,000.
It's 500, about 600 amino acids. So you can multiply that by 3 to get the amount of DNA. But the point is, that looks kind of like, well, what do we do with that? It is a variation of this that's inside the Arctic Tern that implements the quantum mechanical magnetic compass. I've asked biochemists, I said, can you just, if I just said you're going to make a, whatever widget, in this case, I just say hypothetically, a quantum magnetic compass. Could you design, can you give me, is this only 600 letters? Can you give me the 600 letters that'll do the trick? And I actually asked this in regards to ATP synthase, but you could do this for any system. They'll say, no, that's just too hard. That's they know that only a genius that has genius beyond anything of all human capacity could do that. It's that hard of a problem. And it almost makes us, ought to humble us, because that's only, what is that? What the equivalent of 3 chats in the side chat in terms of the letters, but we can't make one like that.
Yeah. Well, and I think the other piece that people kind of forget about is, there are a lot of these folks are arguing that because we're now starting to get some understanding and some predictability of, oh, well, if we see that portion of the sequence, we know it's going to fold like this, right? Like, cool. Just because you know that doesn't mean that it's just going to happen. Congratulations. You know how to tie your shoe. Is it going to tie itself? And that's even dumbed down compared to what you're talking about. You're talking about creating a quantum magnetic compass.
And one thing that Marcos Eberlin pointed out, just citing peer-reviewed literature, this is peer-reviewed scientific literature. They said this outdoes anything any chemist can do in the lab. We've tried. Biology does it better. Where did that knowledge come from? And someone was saying, the birds are trying to migrate from the North to South Pole, and enough of them died along the way And somehow it figured it out. And it's like, yeah, that's not really very compelling. I'm going to share another screen here. And so we've talked about, well, it's not just the fact that DNA can store all this, it's what's stored in it.
Mm-hmm.
Non-coding RNA. This is the title of the article. Computational Mechanisms in Genetic Regulation by RNA. Highlights. Non-coding RNA could be performing computations in ways similar to artificial intelligence systems.
Mm-hmm.
Each RNA species is analogous to a neuron interactions to connection strengths. Instead of genes controlled by on-off switches, they may be regulated collectively. Like neural nets, the computation is fault tolerant, allowing for high mutation rates. This challenges the claim that such RNA is junk simply because it mutates rapidly.
Yep. Well, and they like to argue that, oh, well, if it does something, it's just a template. It's like, yeah.
We're knowing that. And the thing is, just like proteins, every species of RNA can have its own fold and therefore a very different function. DNA is the way we would want it. We don't want it to fold in too fancy a way that will change its basic function of being able to provide sequence information for the genes. Now, I already mentioned it has other utilities, like being a three-dimensional reconfigurable parking lot for molecular machines. But the RNAs are just kind of, they're in a league all of their own. And it's like, okay, why don't you make a DNA system that when it makes RNA, it's going to be a neural net that's going to solve certain problems. Just try doing that. And no human is going to make neural nets of the order that we see in inside living organisms. So
I've actually seen some. I'll find some time. I've actually seen some diagrams, comparisons of the neural net controls in a human cell or human body in general versus, the pinnacle of human tech, and ours are not even close. I mean, it's a joke at this point.
RNAs are almost, I wouldn't say they're not, people ding me and say, well, they're not proteins, but it's like their versatility is, we're only beginning to scratch the surface. And I have a picture here of a link RNA. This is just, let me see if I have it. This is the fire. Oh yeah, the fire RNA. I'm trying to get a diagram up. It's not letting me show the diagram. Man, I'm bummed. See if I can get the fire RNA. See if I can get this figure shown. And it would be lousy. Let me look for another. Just give me a moment here, and I'm going to try to.
While you're looking for that, I'm going to pull some up real quick.
Yeah, go ahead.
Let me if you find it. Okay, so I just pulled up, folks, a little expansion on that same paper. So now they're showing like the process for the data preparation, then being translated from binary, doing direct mapping, Huffman code, rotating encoding, constrained coding. Now it's in genetic code. Then you're doing all sorts of crazy stuff, by the way, folks. And then, doing split into data blocks. It's crazy what's going on here. Then you get into DNA synthesis, or they're using polymerase chain reaction for the random access mechanism. Payload, primer, target, address, file ID, intrafile index, RS code, parity check. It's nuts. Then you're assigning it to a pool. And then you've got the long-term DNA storage. And then if you want to do retrieval, you're doing PCR amplification and DNA sequence to get the data recovery. So consensus alignment, sequence assembly, error correction, decoding, and decryption. And now you're back into binary. It's pretty amazing. And that's looking at, okay, how do we translate it back into electrical impulses so that we can now read it on our computer. It's amazing stuff of what's being being done. But here we go. Sal, this is what I was talking about earlier of them like doing an analysis of a lot of different experiments. And, you know, here they got like magnetic tape, floppy disk, hard drive, flash memory, optical storage, and then DNA storage, and like doing the comparisons of what it can do, what's better at. And then these were actually experiments of different things for data, DNA databases, and what they were able to, so like the stored data size, net coding density, error detection and correction, error-free data recovery, random access. Okay, so interesting. Like in 2012, they weren't able to get random access. To 2013, they couldn't. These guys experiment in 2015, pulled it off. Then 2016, from there, nope, they didn't. They fast forward to 2018, and they were able to do random access. They were able to do error-free data recovery. Net coding density was 1.1 bits per nucleotide. Anyway, the point I'm making is, folks, I don't really know how much more is needed to be showcased of how are they doing direct comparisons to hard drives, floppy disk, flash drive, optical storage, and DNA database storage. How they turn it from binary into quaternary. I mean, I don't know what to tell you at this point. It's obvious.
Now, most of those papers deal with kind of the human side of re-engineering the original DNA. I just needed to point that out.
Well, true, but the we're still using the same medium, and we're using the exact same functions in order to actually store the data, and
not doing it as well.
Well, absolutely. And the thing is that we can program in binary, translate it into raw genetic code, turn it into a sequence, and now have function. We're doing that with synthetic DNA right now, not just for synthetic data storage, but for synthetic proteins that we're trying to create, right? That's exactly what's happening. I mean, they're literally coding the sequence. And I mean, if you can translate something from binary into quaternary and back and forth, I mean, to me, that proves beyond all doubt that we're dealing with code. It's the exact same process. It's not kind of like it, it is. And as you and I talked about before, in that one paper we showed, they had created their own syntax, right? They expanded the symbols for the bases to add for additional data storage types. But this paper right here, augmented encoding characters using degenerate bases. I mean, that's the whole premise of this paper it was published 20th of April, 2019. Let's see, they're getting into, data to DNA encoding. They're using 15 characters instead of 4 to allow for additional encoding characters, shorten the DNA length. You got 15 encoding characters, log to 15 equals 3.9 bits a character. The 4 encoding characters is 2 bits per character. I mean, they're synthesizing degenerate bases. I mean, it's nuts. The reason I'm showing this, because your point, you make, yeah, humans are doing this. But the question you have to ask yourself is there anything that the humans are doing that anybody would ever claim could be done without the human intelligence that's being executed to pull it off? And that's kind of the reason I wanted to showcase this paper.
Okay, I get it.
Is I agree with you in the point you were making. But what I'm showing is, okay, we're talking about the exact same thing, right? We're observing the end result of, in my opinion, the point I'm trying to make is, how does this, what we're researching in genetics right now, in biological systems, we're reverse engineering what the previous genius did, right?
Right, exactly.
Would anybody argue if you came across a genetic sequence that was programmed with a freaking MP3? would you ever argue that it had not required an intelligent agent in order to create the syntax, in this context with a 15-character degenerate system here, to allow for more data store? I mean, all those kind of things. Like, nobody would ever make that argument. I don't think. I mean, I think we'd be laughed out of the room if you did, right? If these guys that are they're working on doing this research right now, if somebody came up and said, like, well, I don't think you're really doing anything special, like natural selection could make that happen, they would probably punch you in the face. Like, you're an idiot. You don't know what you're talking about. How dare you insult me? Are you calling me an idiot? Are you saying I'm dumb? Like, are you insulting my intelligence right now? I mean, I would probably take offense at that, you know? And to me, that's the exact same that's being done in by the YouTube atheists running around and just evolutionary biologists in general, who are claiming that this is not direct evidence of. It's like, would you go and tell your peers that they are nothing special for creating this stuff?
Exactly. Oh, I have my set of diagrams here.
Oh, yep.
So we were talking about the versatility of RNA. I'll show one example. This is, and there's so many things we're discovering. So that it's like saying, well, what do proteins do? There's so many ways proteins can be used. Look in the lower left here, diagram C. There's this link RNA called FIRRE, FIRE. And look what it does. It brings together chromosome 2, a very special part of chromosome 2, another part of chromosome 2, chromosome 17, and the X chromosome. It brings it all together, and it concentrates. The molecular machines will co-regulate these areas because of that link RNA. And that's just one example. There are other examples of the RNA. Now, I want to show what DNA ends up coding for. It ends up coding for proteins and enzymes. And this is a metabolic pathway. This is the glyoxylate cycle here. That's not that important what it is. I'm going to zoom out a little bit, and you can see all the other metabolic pathways here and all the connections. When you zoom out, you'd be dead if this didn't work. You wouldn't be able to digest your food. And I'm just like, yeah, you know, they'll say, well, it didn't build up, you know, all at once. I'm like, okay, how much can you pull out and still be alive? Does anyone figure that out? And so these people are saying metabolism first. I'm like, okay, yeah, this came first, you know, because there's an argument. Is it DNA first, RNA first, proteins first, metabolism first? I say, how about everything first? How come? I mean, that would work, wouldn't it? Except it just looks like a miracle if it were everything first. And I don't know.
For me, when you just showed that, right? So, and this is why, and you know, I've talked about this. Like, yes, I like to argue things like information and code base. You like to argue structural biology and things of that nature. And I'm like, I agree with you. Like, they're all amazing. And then you think about it from an engineering perspective, right? That schematic that you just showed, right? That's probably more complicated than anything humans have ever created.
Oh, you know, there's one area of that, just that little piece of that diagram called the Krebs cycle. That took the guy who figured it out, Krebs, Nobel Prize, but it took half his lifetime.
Right.
Half his lifetime, and it had only a handful of enzymes, and it was just brutal to figure it out. And what's amazing, it's like, okay, why don't you try to build one like that? Not just, you know, not just figure out how it works. Why don't you try to build something of comparable complexity to do something? I don't know that anyone's really building much of an artificial metabolism.
Well, and that is the point I think we need. I'd like to ram home to the audience right now is we don't even understand a minuscule fraction of what is required. Number one. Number two, the arguments that are at this point, things such as it's a code. It's so dumbed down of like, if you can't comprehend this or you're refusing to comprehend this, then there is no way, shape, form, or fashion you're going to be able to understand the higher level, number one.
Oh, yeah.
And then from a macro view of like evidence for God, right? I mean, it's like, Any one of these is enough, right? But we actually have a cumulative case to be made. It's like you've got the structural component, you've got the complexity of the interdependent metabolic pathways as an example. You've got the information component. You've got the, like you have, and all of these in every observed system have to be there. They have to all exist simultaneously in every form of life. You remove any one of them and they're dead. So everything that you guys are arguing are all completely hypothetical, number one. And number two, you're arguing them from a ridiculously dumbed down version. Like, oh, well, you know, it was a super simple version. Like, none of it's simple, dude. Like, it's not simple.
And that's why I don't like these short debates. I said, let's have a 10, 20-hour debate where you can't bluff and just dismiss stuff. And it's in a discussion like that these details come out. And you say every level, one of the levels that are there are at the very high level too, that's complex, like being able to see, being able to hear. And then like the birds, they can actually see magnetic fields, quote unquote. Their eyes can actually see kind of, we try to do simulations as best we can to say, well, this is kind of how its eyes would process this, which is just remarkable that it could, you know, it's like, oh, its eyes can see where the North Pole is. Oh, that's really cool, you know? And he could probably guess his position in terms of latitude based on all sorts of things. And it's like, yeah, these creatures, like, or like a bat it has imaging sonar. I mean, our ears can kind of detect direction, but also not just direction, but distance, that the bat can do this. And sonar operators, and Pigs Can Fly is here. He's been on submarines. I knew a sonar operator, graduate of the Naval Academy. He believed in ID. He said, yeah, I worked with sonar. Whales have sonar. Bats have sonar. Dolphins have sonar. Do you know how sophisticated it has to be to have something that's imaging, that can image position and direction like that? I mean, anyone who builds some of these systems, and because electrical engineers do a lot of stuff with sound waves, well, waves in general, radio waves, sound waves. And then you look at how the ear processes this. One of my professors at the NIH, he was an electrical engineer. It's like, you're doing biology. And I realized that's absolutely the right skill set. And there is something here I need to show you, see if I can grab it. While I'm grabbing it, if you had another point, John.
So, yeah, this stuff, folks, it boggles the mind of, like, all of the things that are required for our basic existence are reaching, like, we're reaching the realization that it's not remotely plausible to conclude we exist through undirected process. And yet we have people who accuse us of God of the gaps, and I don't know, therefore God being the foundation of, you know, the points that we're making. And it's like, no, that is not even remotely the point that I'm making. It's in any other context, would you make the argument, if you had this knowledge, that it's somehow plausible that all this came into existence without the requirement of a intelligent agent in order to execute all of this? I mean, it's just, to me, it's such a straightforward premise that must be acknowledged. Otherwise, you're not actually looking at this from a remotely rational perspective. And I still know how to deal with these folks. It's like we're arguing over, does 1 + 1 = 2? Like on the code argument, that's the way I look at it. Like, you're saying, oh no, it only looks like 1 + 1 = 2. Like, okay, well, if that's your position, I don't know what to tell you. The same thing with structural biology. Like, unless you have the right key, it won't unlock the freaking lock. Like, you have to have the right key to unlock the front door, right? Yes or no? Okay, well, if the answer is yes, and you have to have this exact specific shaped molecule in order to do X. That is the same thing as you have to have key to fit into lock in order to open door. Like, exactly.
I tried to show, I tried to make it accessible. I said, this is the problem of getting things to work in a biological system. Things have to fit. So when we talk about functional folds in proteins, it's like, okay, what do you mean? A lot of the Darwinists want to say, oh, well, we modified a protein and it has a new enzymatic function. I'm just like, you can't generalize that to complex systems because there are many protein systems that are analogous to this little interlocking toy system. The parts have to fit correctly for the complex to work. And so the meaning of what functional fold means is a little different when you consider it being beyond just simple enzymatic function of a monomeric protein. This is seriously difficult to get coordinated shapes that assemble in the right time in the right place. And then there's another thing I want to show. Electrical engineers will recognize this. And I was mentioning my professor of neuroscience. Well, this is actually a description. We use the language of electrical engineering to describe the action of nerves. And you can imagine basically the central nervous system is one gigantic, we might call it VLSI, but that VLSI is puny compared to the central nervous system. In fact, there are more connections in the human brain than there are in the World Wide Web if you talk about the switches and routers. It's just...
And that's in one brain.
And that's one brain. And then in the brain, you have all these little receptors and other things that connect. It's just beyond anything that we can comprehend. And so you mentioned the God of the Gaps thing. I'm like, I personally think God of the Gaps is a good argument. If you got healed, if you're the blind beggar and you got healed by Jesus, I'd say that's a pretty big gap. Of course, people like Matt Dillahunty, he'll say, no, I could be space aliens. Like, okay, what do you do with that? And when I began to hear them openly say, no, they wouldn't believe it. And then you gave the examples of atheists praying for their daughters would all, and then they get healed and they'll just turn around and reject God. I'm like, okay, that's what we're really dealing with. If they will so openly admit that even under those circumstances, they'll reject God. I'm like, well, these other arguments they're surely going to reject. And I just don't deal with them anymore.
In line with what you're saying, it actually is sad. And part of the reason, even though I've been a little bit snarky at times throughout this conversation we've had, I do at the same time have a sadness for the atheist community in general. A lot of academia as well, but I mean the YouTube atheists that we interact with on a regular basis. I feel sad for them because they are having to suspend a huge portion of their analytical ability in order to maintain their worldview, or they're having to refuse to even learn about the things that the other folks are refusing to analyze, right? They're just like, I'm not even going to read about it. I don't even want to know. And because they know if they do, they won't be able to ignore it, right? It's the rational, the logical, and the plausible conclusion if you have the knowledge base and approach it with an open mind. And I mean, at this point, but all in context of the sadness I feel for them is the level of intrigue and curiosity and emotional satisfaction that can be derived from learning about all this new and cool stuff and the new things that we're discovering and things that we've known about for a while that I didn't know about, right? That I'm learning new stuff that we've discovered years ago. I just was unaware of it. The more I learn about it, the more it's like, what? That's awesome. And it puts your entire existence into a new perspective and a new paradigm. And I can't imagine walking through life and operating from the perspective that I am not the result of the pinnacle of genius. But when obviously I also take into account the fall of man and the genetic entropy and different things we talk about. I understand that. It makes a lot of sense. And I'll put this in a very direct context. Today, I was standing in line at Kroger, and there was an extraordinarily beautiful woman in front of me. And as I looked at her, I thought it was interesting. What struck me, it was not only like, wow, she's beautiful, but what really struck me was I can only imagine what the perfect woman would be. Like, as beautiful as this woman I'm looking at is, I can only imagine what Eve looked like, right? And what the capacity for all of because there's the golden ratios. Like, most humans no longer maintain the golden ratio, right? Yeah. We have all sorts of distortion of, like, I'm 6'1", and I've got short arms. It really annoys the crap out of me. I have to get custom shirts sometimes, right? All of us have some random component like that is not perfectly balanced, and we're not at the peak. Heck, I'm shaving my head now. I mean, there's all these different things that we kind of take for granted, that there is a perfection option, right? That was originally what was created. And occasionally when we see these just extraordinarily beautiful people and stuff that have they're pretty close, right? They still have their errors, but they have less than us, right? We got less than you, or like less than the guy who lost his hair, right? And it kind of puts things into context. And I think and what came out of that whole observation I was making was I was more in awe of what the potential that God had created than just the extraordinary beauty of the woman that I was looking at. And what I really thought about as I left was, like, I wonder how often, because I know I sure as heck did never make those kinds of observations before. I still checked out the hot woman, don't get me wrong, and there's no telling what went through my mind in that context, right? But I wasn't thinking about it and having the level of appreciation that I do now based on what I now know and what was required in order for that to happen.
Yeah.
And
definitely.
Anyway, my long-winded diatribe there, I hope that made sense, but it's
it doesn't actually. Jesus said, "Consider the lilies of the valley, how they grow; they neither toil nor spin." But I tell you, Solomon is all his glory was not arrayed as one of these. Now, that has a very different meaning in the 21st century for anyone that's studying gene regulation and photosynthesis and the Calvin cycle and anything else. It's like, oh my goodness, that Solomon in all his glory could not build a lily. Something that's kind of like grass that's here today and gone tomorrow that we mow. Kurt Wise was saying, yeah, once he started knowing about photosynthesis and all the complexity, when he mows the grass, he doesn't look at it the same way. He said, we're just throwing it away. These are examples of supreme design. And we don't even, these are things that we sort of don't give a second thought to. And it's often in the places that you would not think where God's genius is on full display.
To me, I know you've made this argument, I've made this argument, plenty of people made this argument. Per the evolutionary timeline, one of the most exquisite, most complex, and most dynamically mind-bending systems, photosynthesis, was like an early formation. Like, you then argue, when we make the argument about the complexity and such, oh, well, it's had billions of years to get to this point. No. Per your own stupid timeline, one of the most fundamental systems that all these other ones that you claim evolved after it are dependent on. If it's not there, they don't exist, right? You're claiming that one of the most complex evolved way back in the early stages of your own timeline. Explain. And they don't. They just dodge it. And I'm like, okay, please explain the step-by-step process, because we all know that there's multiple components in photosynthesis that, per your own timeline, all existed way back in the day. They all have to be there or photosynthesis doesn't work. So explain the paradoxes of photosynthesis, of all these different subfunctions that are required to work in synchronicity, or you have nothing. expound, and they don't. They just run. And I'm
Math and evolutionary biology don't mix. Math, logic, chemistry, physics, and evolutionary biology don't mix. And that's what struck me. I said, goodness, I mean, I was reasonably competent going through school in all these fields. How is it then that I'm being called out as being stupid that I don't understand? I'm just like, maybe the problem is evolutionary biology doesn't agree with all the other disciplines I've studied. And that's what I see. They run and they get away with it because it's
actually the outlier. It's actually the outlier.
Yeah, it is the outlier, but they get away with it because it's the culture we're in. And part of it is people tend to want to believe that we can figure it out in terms of science and naturalism. I mean, that's just anyone who's in science, you're driven by trying to find a natural explanation for things. And so culturally, even if someone is a Christian, it's like, that's kind of still hard to swallow. It's like, yeah, we probably hit our limit because people don't want to admit there's something that they'll never know and understand and be able to describe. And I got to the point, it's like, well, I don't see why that's so hard, because anyone who's going through hard scientific disciplines and trying to learn, it's like, you realize real fast there are going to be limits to how much you'll ever understand, period. Don't even try.
When you bring that up, it's interesting. One of the things I've really recognized this past month and a half or so is, for the past like 5 months, I've been extraordinarily busy, and I haven't had the time to be reading as much as I had been, the first part of the shutdown. And I was perusing through some stuff the other day because I realized that I'm like, I know I've forgotten some stuff. Like, it's not at the top of mind because I hadn't been paying attention to it as much. And so I've made it a point to kind of start perusing through some of the papers I already had rather than looking for new ones as much. I decided to go back and review some of the other ones that I'd already been looking through. And I'm like, I was reading through and I'm like, oh man, I totally forgot about that. I totally forgot about that. I totally forgot about that. And there were whole portions of thoughts and recognitions that I'd had 6 months ago that, because I kept reading all this new stuff, I had forgotten the significance of some of these other things. And because my mind was being consumed by all these the new realizations, I'd forgotten about the previous realizations and their significance. But at the same time, because of the new stuff I'd learned, I was really realizing and recognizing the even greater level of significance of the first ones that I'd come across. But case in point, to what you're saying about not being able to learn it all, is we don't have the mental capacity to comprehend all this stuff. Now, if we had photographic memories, potentially a different story. And I would argue that used to be the standard, not the extreme rarity.
Exactly.
And when you think about that, it puts human existence into a whole nother perspective. And to me, the premise of, and I've made this argument before, of, okay, let's think about this for a second. Let's suggest for a moment that humans could live to be, say, 500 years old, right? And you have photographic memory. Think about, all the things you could learn, your realization, your comprehension, number two. But then let's put this in real world context. Like, let's say that Galileo is still alive now, and Newton, and Copernicus, maybe? I don't know. Einstein, Heisenberg, all those, like, they're still alive right now. They're all alive. What would Newton know now? Right? What would he be being able to download into Einstein's brain? Right? Because right now, Einstein would be the young buck. Right? He'd be the young guy compared to a Galileo and Newton. But when you really think about we talk about the premise, I think Newton actually coined this term of, like, I stood on the giant's shoulder of the giants who came before, right? Well, think about that in the context of the giants were still alive, and how much more accelerated our ability to create new technologies, have greater understanding of the universe, things of that nature. But the ultimate point I'm getting at is, yeah, that'd be awesome. But we would just be able to have greater understanding of the genius of he who made it possible.
Yeah.
And where I'm going with this ultimately, my ultimate conclusion of all this is, I honestly wonder if our incredibly short timeline of existence actually mitigates our ability to truly comprehend the awesomeness of it. Like we don't have enough time. Even if we were focused, we don't have enough time to truly comprehend the point.
Well, the Lord has compensated for that because we have technology to help us. We have researchers that record this and they can tell us. And we have enough, just like in the days of Newton, they didn't have radar and understanding of magnetic navigation like we do now. So we thankfully have in the 21st century a perspective that is very special, that we can Google things and learn. And so it's not like the Lord has left us without any witness. In fact, I've argued this is really miraculous that even in our degraded condition relative to our ancestors, we're able to make more inferences in the 21st century about the existence of God than at any other time.
Sure.
And so we can still be, the Lord has really provided, despite ourselves, to comfort.
It is ironic that it's now measured that we're getting dumber.
Yeah.
I mean, average IQ has been on the decline. They've been measuring it for the last 100 years. It's been dropping like 8 points a decade. I mean, think about that. It's been dropping 8 points a decade. That's 1 decade. 8 points average. That just one.
That seems a little high. I'm not going to. I'm just a little cautious about that number, but we are dropping. And in my debate with Dapper Dino, I cited that study. I say, look at all the intelligent women. They're more likely to be childless. I said, look what natural selection's doing.
Mm-hmm.
And I think it's getting lost on a lot of people. They're just trying to evade the problems of genetic entropy. It's like, guys, name one major geneticist that thinks our genome's improving. That's a start. And
Dan
then you
Grauer.
look.
And ladies and gentlemen, for those of you who don't know who Dan Grauer is, he is one of Sal's personal irritants. And he claims that the whole genome is junk.
He's, I guess I could say this, he's a loser. I mean, people don't even want to, he's not even publishing anymore. It's like, what does he have to say that's of meaning to the medical community? He'll just say, you guys are idiots, which he did. He called the ENCODE consortium of the NIH and by implication, Francis Collins, ignoramuses and crooks. I'm just like, you don't say that to these guys that are at MIT, Harvard, Yale.
At the time, wasn't
I'm like, what
Collins
kind of
the head
idiot
of the
are you that you're going to say stuff like that? You're just like an internet troll.
At the time, wasn't Collins the head of the NIH at the time?
Yeah, he called Collins the creationist head, just vitriol. I'm just like, dude, no one wants to listen to you. You're not making any contribution to advancing medical research. These guys are, and you're calling them idiots, crooks, and ignoramuses. Calls them crooks and ignoramuses. It's the scientific equivalent of Saddam Hussein. I said, what good are you as a scientist? What contribution has someone like you, an evolutionary biologist, made to the advancement of science and our understanding of biology? Zero. And so I have a pretty low opinion of him as a scientist and as a human being. Zero contribution. He's just like these internet trolls that just whine all day, make no contribution to advancing our understanding. So,
and I think one of the other key points too about that type you're referring to is it is becoming blaringly obvious that a significant percentage of them spew their talking points without even actually listening to the argument that they're responding to. It's just, I'm going to just blanket dismiss whatever you said. Insert insult, standard talking point, close quote. And it's like like case in point, okay? If you go on MDD right now, you will find comments talking about, oh, just another theist spewing this it's a code argument. John Perry is an atheist, said he was an atheist, reiterated that he was an atheist, and also was trying to like downplay the theist argument throughout the whole debate. So why then, if you actually watched the debate, would you be claiming that it's just another theist talking point, spewing talking points? Oh, wait, you didn't watch it. You're the one spewing talking points. You are literally projecting your own stupidity and your own ignorance. It's amazing. And the most fun I've had from those comments, by the way, is this one guy, Blue Heron, I think his name is. He's been talking. He called talking trash.
Yeah, Blue Heron.
He talked trash about me for like use my name, and I'm like, John Perry was literally using the exact same arguments that people have heard me make in debate after debate after debate, almost verbatim. And you're claiming that his are normal or like are more substantive and he understands the process, all that kind of stuff. While simultaneously you have been, for months, you've been saying when I make the exact same statement that it is false and out to lunch. So which one is it? Which one is it? It's a
and Blue Heron, just another biased critic. I was talking about fitness and he said Sal doesn't know anything. I cited all these papers. I talked about coupled differential equations and the problems for fitness. Did that like fly over your head? And I'm thinking, yeah, it probably did. Sometimes the argument's too sophisticated. These people don't understand and they just presume I'm dumb. You know, it's like maybe if they'd listen more carefully, which, you know, the only reason I would even engage these people is if I get invited and there's an advertising opportunity. But I'm investing my time now with Christians who are students in the sciences, and because they had many of the same struggles I had, and they may be in science classes, they're outnumbered 100 to 1, or may feel that way. They may not be actually outnumbered that much, but you don't realize it. That you may think that you're really badly outnumbered. And there are probably a lot of students that are also sympathetic to your views about design. And these are the people I'm going to be investing time with. You know, I've unsubscribed from like Speed of Sound's channel. It's like, you know, I'm just not going to waste time, you know, but since he offered to do independent verification and validation, I might accept that. Now it's going to be funny if he doesn't go actually follow through with his promise. I mean, not his promise, but what he's claiming. And I'm like, okay, yeah, we'll see if he can do a simple task. A simple task that maybe a first-year college student could do. Let's see if he can draw a plot with his spreadsheet that he makes on his own. Is he going to be able to do that and say, Sal was right? Look at that. I got that slanted graph just like Sal did. We're going to find out if he's going to follow through. And if he doesn't do that, he's not worth my time anymore. Serious.
Well, I'll tell you this much. So he has been whining for 6-7 months now at me. Okay. And I have stated multiple times, and he said he was going to do it multiple times. I'm like, create a video, outline your objection to my arguments, send it to me. I'll respond. Not some long-winded freaking 3-hour thing. Like, create a video specifically, like bullet points. Here, this is my. Here's your point. Here's my point. Here's your point. Like, and I will respond. I've told him I will respond if he does so. Hasn't happened, and we're going on like 7 months now, and then he continues to whine and say that I won't ever engage, and I'm like, I have literally made the public offer, just do it, and I'll respond.
Well, you know what he did on his 70th birthday? He wrote me a letter kicking Sal's ass, and invited me to his StreamYard while Rob Stadler was talking to join all his haters. I'm just like, okay, this is how you spend your 70th birthday. You write a nasty letter like that. I never said, up until tonight, I never had a harsh word for him. And yet he's been trash talking. And it finally got up to here, you know? It's like, you don't do that to friends.
Well, true.
And so I'm just like, I'm done, you know? I'm done. and he'll come on people's channels and just start insulting them. I'm just like, okay, I don't do that on his channel.
Yep.
And I'm just like, yeah, I'm done wasting time. The only reason I would engage the critics is that they're going to do independent review and say, they'll find some errors and correct my work. And in the process of him asking me to do this, I did find one little math error corrected. It wasn't material, but now I have a better presentation. I have better calculations. And let's see if he's going to be able to. I gave him a 3-minute recipe. It's not that hard. Let's see if he's going to publish on his website a little slanted graph with some dots. Okay. I don't know if I wouldn't take a bet on this because I don't know what he's going to do. But it's just going to be ridiculous if you can't do that, because I basically spoon-fed him the formula. Said, if you can't put that on a spreadsheet yourself, I'm not going to, sorry, I'm just blowing off some steam because it did get under my skin.
It's very
But
well
he would use his
justified.
email to say, kicking Sal's ass on his birthday. I said, where did that come from?
This is
Where
why
did that come from? I didn't incite him to say junk like that toward me.
this is
He's just
why
getting irritated now. And it's like, okay, if you don't like the arguments, don't have to take it out on me. You can refute them. You can do the math. If my math is wrong, he can refute the math.
that is why
just low.
I've stopped engaging with there's multiple, not just him, but there's multiple I've just kind of stopped engaging with. I'm like, if you're not going to have any kind of like actual conversation, an adult dialogue, then I'm going to treat you like I treat you, right? Like, you want to come in and spew talking points and insults. I fight fire with fire, bro. Like, let's go. I'm not gonna. One of the biggest issues I have with where things have, and I'm speaking in a general sense, right, of the quote religion, right? Christians have conceded and conceded and conceded for the last, I'd say 50 years. And where are we now? Like, oh, we just talked nice. And oh, well, we're just going to tolerate this and all that. Like, we're not going to fight back. We're not going to put things into context. We're not going to like argue our case. And I'm like, and then we wonder why we're where we're at. And I'm like, I'm not going to run around claiming to be a statesman. That's fine. It's if we're now in the equivalent of the street yard brawl, so be it. But we're undergoing genetic entropy, societal entropy, all aspects of our existence are collapsing around us. And you know what? If I got to fight back, so be it. And now, is that the style for everybody? No, but I'm okay with being that character in the play, and it's just something's got to be addressed, in my opinion. And if we don't, then I refuse to go quietly into that good night. As the by the way, that's one of my favorite poems of all time. Is the go not quietly into that good night. Although I like to say, I shall not go quietly into that dark night. I think it's better than the good night, but that's just me. Okay, so Sal, I'm thinking here in a couple minutes you and I wrap up, and audience, I'm considering doing a short open mic stream, but I do not want to do that in conjunction with this stream. So I think this is a great topic between me and Sal, and a good conversation. So I'm thinking about wrapping this one up here in a couple minutes, and then maybe do another stream. So who in the audience is up for quasi after show to this discussion? Let me know in the chat, and think about doing that. What are your thoughts, Sal? What do you think about this overall context of what we've been talking about tonight?
Well, I'm just glad to have the chance to talk, and thanks for the free advertising. And I just want to alert the viewers that there's going to be a talk on Sunday night on there's going to be a talk on Sunday night on eukaryotic evolution, and you'll see another levels of complexity. We'll talk about how the complexity, the nested hierarchy, does not explain it. One would have to be invoking non-random mutations to explain eukaryotic intracellular, the evolution of intracellular transport in eukaryotic organisms. So, so that's basically all I had. I'm kind of running out of gas, so that's probably why I didn't have any other thoughts. But the complexity, I'm glad I had a chance to talk about it, and thanks for the interesting papers that you found on some of the DNA storage that humans are attempting.
Yeah, all of those things. I mean, like as we've been talking about tonight, when you kind of when you put it all into context, it creates just an entirely different paradigm, in my opinion, that has to be addressed. And yeah, like the standard term is like the conventional paradigm, right? I'm like, the conventional paradigm is a joke at this point. And you know, we listen to people, oh, well, we're already accounting for these things with extended modern synthesis. And I was like, no, you're not. No, you're not. Get out of here. No, you're not. I'm sorry. If you are, then you're doing it on like level 1. And we're already up to, with the, you know, as you talk about on a regular basis, the dramatic expansion of discovery that's happening. It's an exponential snowball. And it's like, so you, dude, you're way back at the top of the hill. We got an avalanche going down, you know, and you're claiming, you know, the avalanche is underway and you're saying there's no avalanche. I mean, oh no, all the stuff that's happening in the avalanche has no bearing on what I'm doing. Well, yeah, that exactly, because you're not engaged in it. And that you're telling all the other people who are that they don't know what they're talking about. That's what's happening. And I find it fascinating to observe. And just you know, to put into context, and I strongly recommend everybody make sure you make it over to Sal's stream on Sunday. I'm sure it will be epic. And for entertainment purposes, you might not make it all the way through the 55-minute video that Sal's got, because I imagine it gets pretty complex. But
Well,
the 3-minute version,
yeah,
is
I'm going to take
well worth it.
I'm going to take it down temporarily because Speed of Sound complained that I used his real name. I'm just like, well, you came on my channel as Mike Bruzenak, but I'm not going to give him an opportunity. So I'm going to take it down temporarily. I'm going to redo it just in deference to him. But he and I are done.
Well,
you know, unless he's going to seriously just try to recapitulate the plot with what I've given him, he's not worth my time. And I'll try to avoid using his real name because I don't want any accusations that I was trying to, you know, out him or anything. I thought it was, so that'll be it. We're done. You know, right now he's just trawling and trying to cause trouble. I may block him from my channel. I just don't need that crap anymore. I have to focus on molecular biology, chemistry, cell biology, organic chemistry. There's a lot on the plate. And then the stuff that, you know, all the other stuff and studying complexity. I don't have time to be wasting on trollish behavior. I really don't.
Yep. Well, in that light, folks, we're going to wrap things up. Appreciate everybody coming, paying attention, having input, and having a great side chat conversation. I'll probably post a link to the folder, my Google Drive, that has the papers that we were kind of breezing through. I'd recommend looking through those. I mean, it's some mind-bending stuff. If you like technology, your mind will be blown at what's going on. But come on over, join us in the after show. I just posted a link in the side chat. I'll put the join the stream link in there in the other chat on the other page in just a minute. But let's do that. But as I love to say, as you contemplate the things that we discussed tonight, their implications, their ramifications, and attempt, if you will, to look at the cumulative case that was being showcased and everything that we talk about. I mean, Sal's showing crazy schematics of metabolic pathways, and we're talking about code, and we're talking about 3D structures and locks and keys, all these different things. Just think for a minute, take a pause, time out, breathe, and rationalize to yourself, is the conclusion you have reached about the very foundation of your existence a logical, plausible, and probable conclusion, or are you just ignoring the obvious? Thanks so much, y'all. Y'all have a great night, and we'll see you on the after show.